Endocrine regulation of muscle development and growth is a complex and intricate process involving multiple hormonal pathways and interactions. Skeletal muscle, composed of contractile fibers and supporting connective tissue, plays a crucial role in locomotion, posture, and energy metabolism. Understanding the anatomical considerations of skeletal muscle and its components is essential to grasp the intricate hormonal control mechanisms that govern muscle growth. In this script, we will delve into various hormonal factors that influence the development and maintenance of muscle tissue, including growth hormone, insulin, testosterone, estrogen, cortisol, thyroid hormones, insulin-like growth factors, myostatin, adrenocorticotropic hormone, glucocorticoids, and anabolic steroids. Skeletal muscle is a complex tissue composed of a hierarchy of structures. At the macroscopic level, muscle is organized into groups of muscle fibers called fascicles. Within each fascicle, individual muscle fibers are enveloped by a connective tissue layer called the endomysium. Fascicles themselves are bundled together by another layer of connective tissue called the perimysium, forming larger units known as muscle bundles. Finally, multiple muscle bundles are enveloped by the epimysium, which provides structural integrity and connects the muscle to tendons. Within each muscle fiber, myofibrils run in parallel. These myofibrils contain the contractile units of the muscle, called sarcomeres, which are responsible for generating force and facilitating muscle contraction. The sarcomeres consist of overlapping thin actin filaments and thick myosin filaments, organized in a highly ordered structure. The functional unit of muscle contraction is the motor unit, which consists of a single motor neuron and all the muscle fibers it innervates. Motor units enable precise and coordinated muscle movements by varying in size and recruitment patterns. Understanding these anatomical considerations provides a foundation to explore the intricate hormonal control mechanisms underlying muscle growth and development. Muscle growth and development are regulated by a complex interplay of hormones and factors that act in a coordinated manner. The primary hormones involved in muscle growth include growth hormone, GH, insulin, testosterone, estrogen, cortisol, thyroid hormones, insulin-like growth factors, IGFs, myostatin, adrenocorticotropic hormone, ACTH, glucocorticoids, and anabolic steroids. These hormones exert their influence on muscle tissue through various mechanisms, including protein synthesis, protein breakdown, satellite cell activation and proliferation, and the regulation of muscle fiber type. Understanding the role and interplay of these hormones provides valuable insights into the physiological processes that drive muscle adaptation to exercise, development, and aging. Growth hormone, GH, is vital for muscle development and growth. It is primarily synthesized and secreted by the anterior pituitary gland in response to hypothalamic releasing hormones, specifically growth hormone releasing hormone, GHRH. GH acts on target tissues, including skeletal muscle, through its interaction with the GH receptor, which subsequently triggers a series of intracellular signaling cascades. GH exerts anabolic effects on muscle tissue by stimulating protein synthesis, enhancing amino acid transport into muscle cells, and promoting the proliferation and differentiation of satellite cells. Moreover, GH promotes lipolysis, which supports energy supply for muscle anabolism. The pulsatile secretion of GH and its control by various physiological and pathological factors play essential roles in the regulation of muscle growth and development. Insulin plays a crucial role in regulating muscle protein metabolism. It is secreted by pancreatic beta cells in response to elevated blood glucose levels. Insulin acts on muscle cells by binding to insulin receptors, initiating a signaling cascade that promotes glucose uptake, glycogen synthesis, and protein synthesis. Insulin stimulates muscle protein synthesis by activating the phosphoinositide 3 kinase/protein kinase B/mammalian target of rapamycin, PEEK/ACT/mTOR pathway, which enhances translation initiation and ribosomal activity. Insulin also inhibits muscle protein breakdown pathways, resulting in a net gain of muscle protein. Moreover, insulin promotes nutrient delivery into muscle cells, allowing for the replenishment of glycogen stores and supporting muscle anabolism. Testosterone, a primary male sex hormone, has potent anabolic effects on muscle tissue. It is produced by Leydig cells in the testes and, to a lesser extent, by the adrenal glands in both males and females. Testosterone binds to androgen receptors in target tissues, including skeletal muscle cells, where it modulates gene expression and protein synthesis. Testosterone promotes muscle hypertrophy by enhancing muscle protein synthesis and inhibiting muscle protein breakdown. It also increases satellite cell activation and proliferation, contributing to muscle repair and growth. The anabolic effects of testosterone on muscle tissue contribute to sexual dimorphism in muscle mass and strength, as observed between males and females. Estrogen, the primary female sex hormone, plays a role in muscle mass and strength regulation. Estrogen is primarily produced by the ovaries in premenopausal women and by the conversion of androgens in adipose tissue through the action of aromatase. Estrogen acts on target tissues, including skeletal muscle, through estrogen receptors. It modulates protein metabolism by regulating protein synthesis and breakdown pathways. Estrogen can indirectly influence muscle growth by promoting fat deposition and modulating nutrient availability. Additionally, estrogen enhances glucose uptake into muscle cells, contributing to energy metabolism and muscle performance. The effects of estrogen on muscle mass and strength are complex and involve interactions with other hormones and factors. Cortisol, a glucocorticoid hormone, plays a crucial role in muscle protein metabolism. It is primarily released by the adrenal cortex in response to stress, exercise, and circadian rhythms. Cortisol acts on muscle tissue by binding to glucocorticoid receptors. It induces muscle protein breakdown, leading to the release of amino acids into the bloodstream. Cortisol also inhibits muscle protein synthesis and promotes the oxidation of amino acids. The balance between cortisol and anabolic hormones, such as testosterone and insulin, is critical for maintaining muscle protein homeostasis and adapting to stressors. Thyroid hormones, including thyroxine, T4, and triiodothyronine, T3, regulate muscle metabolism and energy expenditure. They are synthesized and released by the thyroid gland in response to thyroid-stimulating hormone, TSH, from the anterior pituitary gland. Thyroid hormones regulate muscle protein metabolism by modulating protein synthesis, degradation, and turnover. They influence muscle fiber type composition, with higher levels promoting the transition from slow twitch, type I, to fast twitch, type II, muscle fibers. Thyroid hormones also regulate mitochondrial biogenesis and oxidative metabolism in muscle cells, impacting energy production and utilization during muscle contraction. The interplay between thyroid hormones and other hormonal factors, such as insulin and cortisol, influences muscle growth and adaptation. Insulin-like growth factors, IGFs, are peptide hormones that interact with insulin receptors and exert anabolic effects on muscle tissue. They are synthesized and released primarily by the liver and other tissues in response to growth hormone stimulation. IGFs, mainly IGF-1, mediate the anabolic actions of growth hormone on muscle cells. They promote muscle protein synthesis, inhibit protein breakdown, and stimulate satellite cell activation and proliferation. IGFs also enhance nutrient uptake and utilization by muscle cells, further supporting muscle growth and development. The intricate regulation of IGF signaling and its interplay with other hormonal factors modulates muscle growth and adaptation. Myostatin, a member of the transforming growth factor beta, TGF-beta, superfamily, plays a vital role in muscle development and growth regulation. It inhibits muscle hypertrophy by negatively regulating muscle protein synthesis and satellite cell activation. Genetic or pharmacological inhibition of myostatin has been shown to result in significant muscle hypertrophy in animal models and human patients. Inhibition of myostatin signaling increases muscle fiber size, satellite cell proliferation, and muscle protein synthesis. Exploring the mechanisms and potential therapeutic applications of myostatin inhibition provides insights into enhancing muscle growth and treating muscle wasting conditions. Adrenocorticotropic hormone, ACTH, is secreted by the anterior pituitary gland in response to corticotropin-releasing hormone, CRH, from the hypothalamus. ACT stimulates the release of cortisol from the adrenal cortex, influencing muscle adaptation to stress. ACT modulates muscle protein metabolism by regulating protein synthesis and breakdown pathways. It also affects muscle fiber type composition, energy metabolism, and immune function. The interplay between ACT, cortisol, and other hormonal factors influences muscle adaptation to physical and psychological stressors. Understanding the role of ACT in muscle physiology provides insights into the mechanisms underlying stress-induced muscle changes. Glucocorticoids, including cortisol, and anabolic steroids influence muscle physiology through their interactions with glucocorticoid and androgen receptors, respectively. Glucocorticoids, when elevated, promote muscle protein breakdown, inhibit protein synthesis, and modulate muscle fiber type composition. Chronic exposure to high levels of glucocorticoids can lead to muscle wasting and weakness. Anabolic steroids, such as synthetic derivatives of testosterone, have potent anabolic effects on muscle tissue. They enhance muscle protein synthesis, inhibit protein breakdown, and promote muscle hypertrophy. However, the use of anabolic steroids for non-medical purposes can have detrimental health effects and is prohibited in sports. Understanding the effects of glucocorticoids and anabolic steroids on muscle physiology highlights the delicate balance between anabolic and catabolic influences on muscle growth and function. In conclusion, the regulation of muscle development and growth is a complex process influenced by numerous hormones and factors. Anatomical considerations of skeletal muscle provide the foundation for understanding the intricate hormonal control mechanisms involved. Growth hormone, insulin, testosterone, estrogen, cortisol, thyroid hormones, insulin-like growth factors, myostatin, adrenocorticotropic hormone, glucocorticoids, and anabolic steroids all play critical roles in modulating muscle protein synthesis, breakdown, satellite cell activity, and fiber type composition. Exploring the interplay between these hormones provides valuable insights into the physiological processes underlying muscle adaptation to exercise, development, and aging. Further research into the intricate mechanisms involved may pave the way for targeted interventions to optimize muscle growth and function and combat muscle wasting conditions.